Hello, my name is Chris Peterson, and I'm partnered up with Jacob O'Connor and Kedra Osea. And this here is our presentation on Figure 3C from Murray and Kirshner's journal article titled Cyclin Synthesis Drives the Early Embryonic Cell Cycle. This here slide contains all the research questions, dash objectives, from both the figure and the overall research paper. The questions from the research paper, well, there are quite a few, and they all consist of what initiates the division processes such as mitosis and meiosis, mainly mitosis, in cells in the early embryos of xenopus frogs, which are also known as African clawed frogs. Is it a molecular factor that drives these processes? And also, if so, how does the concentration of these molecular factors affect the rate at which these cells divide? Also, another important thing to consider is how do inhibitors of the mRNA, which of course serve as a basis for protein synthesis, affect the rate of the processes? And also, how do factors controlling mitosis in early embryo extracts differ from other parts of the body, such as, the somatic areas with somatic cells? However, in the figure, it's much simpler, obviously, does adding cyclin mRNA affect the rate of mitosis in xenopus embryo extracts? If so, how long does it take for a cell to transition from interphase to mitosis and then revert back to interphase? So here are the other figures. They serve as a basis for understanding the one we went over. Figure 3a shows the patterns of protein synthesis and the process of mitosis in a mock-treated sample and a sample that has been treated with RNases, which are enzymes that pretty much break down RNA. The mock-treated sample had dark bands, as you can see right here. And it's been observed that nuclear breakdown occurs at about 30 minutes, while telophase occurs about 40. However, the one that actually has been treated, as you can see, has no bands at all, meaning it did not undergo any protein synthesis, nor did it undergo mitosis. 3b pretty much gives us a comparison of how the differing concentrations affect the rate of these processes. As you can see here, an increased concentration causes the rate of mitosis to increase. The first and highest concentration was 5 micrograms per milliliter. And it was observed that nuclear breakdown occurred at 40 minutes and 120 minutes when it underwent a second cycle, and telophase was reached about 50 minutes. But as you can see here, when it gets down to like 2.5 or 1 microgram per milliliter, nothing happens at all. No nuclear breakdown or no mitosis. This is our experimental rationale and method slide. Pretty much, it was a really good choice for them to use xenopus because the embryos extracted from the xenopus frogs, they actually develop outside the mother, so they're much easier to extract and, of course, make samples out of. And their embryos have also been observed to easily be manipulated by chemical treatments, such as like proteins and RNA. And of course, that makes it easier to get your results when you're researching about things like the cell cycle or signaling transduction pathways during the developmental process of the embryo, or embryonic induction. 2 different samples were used in the experiment represented by figure 3c. One with the added cyclin mRNA with a concentration of 5 micrograms per milliliter, because that's the only one observed to actually undergo mitosis, and one without it. And this was done to make a comparison to see if cyclin mRNA was sufficient enough to induce the mitosis. And researchers got an insight into this process to determine whether or not cyclin mRNA is enough. They used fluorescent images at different time periods, at different points of time, to see what happens, to see when a cell is in interphase or mitosis. All right.
In the methods section of this experiment, the cyclin abundance was measured using densitometry. This measures the optical density of light using light-sensitive materials, for example, a photographic film. This is done using a densitometer, which records the fraction of incident light transmitted. However, while looking in the method section of this experiment, the unit usage was not continuous and the concentration of RNase inhibitor was not given and therefore was inferred to be the same units used for the incubated RNase in the beginning of this experiment. While looking at the flowchart, this experiment starts with a incubated cyclin extract of pancreatic RNase at a concentration of 0.25 micrograms per milliliter, which is incubated at a temperature of 10 degrees Celsius for 20 minutes. After which, 0.05 micrograms per milliliter of placental RNase inhibitor is added to the sample, which is then incubated for an additional 10 minutes at 10 degrees Celsius. After which time, there is added 50 micrograms per milliliter of calf liver transfer RNA, S-methionine, at a concentration of 0.4 per milliliter, and sea urchin cyclin B mRNA and 100 millimolar calcium chloride and 1 millimolar magnesium chloride. After this process was done, the mRNA was transcribed by the T7 polymerase from the sea urchin cyclin B cDNA clone. Once the completed experiment was done, it was treated with an RNase-free DNase to destroy the template DNA. Once the two cycles were completed, the one with the concentration of 5 micrograms per milliliter and one with the sample with no added mRNA, looking at the data shown, the sample with no added mRNA did not run while the sample with mRNA completed 2 cell cycles. It took the sample with added mRNA 20 minutes for the cell to transition from interphase, and then it took an additional 20 minutes for it to go from interphase to mitosis, after which it took an additional 30 minutes to get from mitosis back to interphase, and then another 50 to transition from interphase into mitosis for a second time, therefore completing 2 cell cycles of mitosis and a total of 120 minutes. The sample with no mRNA did not complete any mitosis cycles at all.
And as you could see below, we have the picture of the figure right here, which visualizes or gives us an insight into the cell cycle that was actually completed twice when the RNA was actually added to the sample. Here, go back. As you can see, the pictures are kind of inconsistent, but as you can see, 20 minutes, the cell is in interphase, but 40 minutes undergoes mitosis, and 70 minutes, you can see it's in interphase again. You see no nuclear breakdown occurs, and mitosis happens at 120. We assume that the bottom picture represents the sample without the added RNA, and therefore did not undergo any type of cell division process. That's good.
So for the conclusions, the results of this figure conclude that the sample with the 5 microgram per milliliter mRNA was able to undergo mitosis and transition back between interphase and mitosis across the 120-minute total time period, whereas the sample without the added mRNA did not complete any cell cycles and remained unchanged. The concentration of mRNA is still sufficient to start the transition from interphase to mitosis at a concentration level of 2.5 micrograms per milliliter from interphase to mitosis, although the overall time to transition was longer. This provides that the presence of cyclin mRNA is sufficient to induce the process of mitosis in the embryos extracts from xenofish frogs. We can support the fact that the presence of mRNA in the samples is enough to drive mitotic processes because it reduces the activator for the process, but there was no clear evidence for the effect that no additional mRNA has on my mitotic process. Some personal critique and feedback that we had for this article. The main problem with this figure was that there were a couple of images on the bottom that had no labels, which made it harder for us to determine what the images represented. We believe that it most likely represents how the sample without mRNA does not undergo a reaction after RNA has been added. We also noticed that the first two images seem to have been unintentionally swapped, since the rest of the images have the black background representing the mRNA, while the gray fuzzy background represents a phase in the cell cycle at a particular time. For the first two images, however, the one with the black background apparently represents the cell in interphase, while the one with the gray background represents the cyclin mRNA. Although a very small change, it does present the reader with some confusion and can lead to greater confusion when trying to analyze the article. Some improvements that can be made to this experiment and journal consist of adding a label to the unlabeled photograph in the figure, providing more evidence of how no additional mRNA does not induce a mitotic process, and taking pictures at different times to further prove this trend, and conducting experiments on how different concentrations of cyclin mRNA affect the rate of mitosis if they start it all, and providing pictures taken at different times. For our references, we have here listed the main journal article from Murray and Kirschner, and then an image link that we used in the previous slide. Thank you.
In the methods section of this experiment, the cyclin abundance was measured using densitometry. This measures the optical density of light using light-sensitive materials, for example, a photographic film. This is done using a densitometer, which records the fraction of incident light transmitted.